Hi, everyone. So I'm Bérénice Batut. I work for the French Bioinformatics Institute, and I will talk about... Oh, the mic, sorry. Oops. It was just to be sure that you are awake. Good. So I'm Bérénice Batut. I work for the French Bioinformatics Institute, so in France, and I will talk today about the Microbiology Galaxy Lab and the effort we put together with the microGalaxy community. So, about microbiology and microbial data analysis. So we ran a survey in 23 where we asked people in the research, microbiology research community, what are they doing? So what are they interested in? And they are mostly interested in bacteria, eukaryotic, so they study this type of organisms, but they're also interested in mixed communities, like microbiome. And which technique do they use? It seems they use a lot of single organism studies, so they take one organism and they sequence it, they study it, but they do a lot of metagenomics, also metatranscriptomics analysis. And which type of analysis are they doing with this data? So they are interested in functional analysis, they are interested in identifications of genes, assembly, comparative analysis, a lot of different things. So it was good for us to know what are the scopes of the people, what they are interested in. And when we asked them if the tools for their analysis are available on Galaxy, will they use them? And 70%, more than 77% of the people say yes. So that's like, yes, great. If we get the things in Galaxy, that could be useful for them. And the question is, could really scientists nowadays use Galaxy for microbial data analysis? The answer is, over the years, the number of tools available in Galaxy for microbial analysis has been just increased over the time. If you see, that is the blue line here. So we have a lot of tool suites available for microbial analysis. And it's the same for tutorials. So if you see in the orange curves, we have more and more tutorials available that cover microbial analysis. And in red, this is workflows. That has been increased a lot over the last few years. We have more and more workflow available for microbial analysis. And they are really used. So if we look at how many users, so here are the number of users for the last 5 years on the use Galaxy.star servers, and here the number of times the runs have been done, and you see that they are used, and you have tools like Kraken here that has been used by almost like more than 50,000 individual users, more than a lot of times here. And if we see that the more the tools are included in tutorials and workflow, the more they are used. So it's something we saw when we looked at the data. And the resources available for microbial analysis are covering a lot of different types of analysis. So this is in our operation, and we looked where are the tools and how they are used in terms of switch runs, switch users. And we have like basic sequencing, quality control, trimming, formatting, but we have also genome annotation, a lot of antimicrobial resistance prediction that has been used over the last few years, but also, assembly and everything there. And if we try to represent that in a more structured way, we cover with Galaxy all the type of analysis you could think about for when you are interested in microbial data. So if you have a microbial isolate, so for one species or one organisms, and you are interested in genomics, transcriptomics, or proteomics, here are the different type of analysis you could run. And every small numbers here are pointing to the list of tools available for each of these steps. So we have a lot for each of these different type of analysis. We have tools available in Galaxy for that. And it's the same for microbiome analysis. So we do exactly have the same type of analysis. We can do, if we are microbiome metagenomics data, we can do antibiotic resistant gene predictions directly from the reads. We can build metagenome assembled genomes and build the gene catalogs from that. So we can do a lot of things and use this catalog afterwards for metaproteomic analysis. So that are the type of things we can do in Galaxy. But do scientists really use Galaxy for publications? So not just, okay, hypothetically, we have a lot of tools, but are they using it and do they cite it? If we look at the publications over the years of the Galaxy publications, and we try to extract only the ones that are related to microbiome, we see that 28% of the publications citing Galaxy are microbiome related ones. And they cover either ecosystems, dynamics and biodiversity, health and disease, or both topics. So that is all this area here. And you see that it's growing like it's growing the number of publications over the years. And if we look at several examples, we have a lot of different small examples of interesting studies that they run. So for example, they looked at the Klebsiella pneumoniae and several strains of them that are antibiotic resistant. And they put it in under different treatment with different antibiotics. I will not put read the names, just, yeah, I don't want to make a mistake on that. And they did a phylogenetic and genetic analysis of Klebsiella pneumoniae within Galaxy. So that is an example of papers where they use Galaxy, and it was published 2 years ago. Another type of analysis that has been run using Galaxy is a study of cloud microbiome. So they went on the top of a volcano where they collected microbiome within clouds and without clouds, and they compared the microbiome community there on the top of this volcano. And they could look, so we were participating in that with NG there. And so we could look at the bacteria communities inside and the eukaryotic communities inside. And they collected not just metagenomic data, but metatranscriptomic data also. And we could look at which pathway are differentially expressed between cloud and clear atmosphere. Another type of analysis that has been run is building from metagenomic data, building metagenomic assembled genomes and being able to reconstruct full pathway and using that to map metaproteomic data on that and to identify on this pathway, on this glucolase pathway, which are how they are expressed, which proteins are expressed and which abundance along this pathway using metaproteomic data. And the final example, so using fecal metatranscriptomics, they could compare biochemical pathway in traumatic brain injury patients and control patients and looked at which pathways are differentially expressed between these two cases. So this is just a few examples of different types of study that has been run using Galaxy. But, okay, they're using it Galaxy, but can we do something even more to help even more people to use Galaxy? And it's where we came and started to build the Microbiology Lab. So you have heard maybe about subdomain in Galaxy. The lab is a new structured way of building subdomains of a Galaxy server with a custom landing page and toolbox. And it's rendered using the lab engine that has been developed by the Australian team. And so here you have a lab specific tool panels view here, and then the central panels that you can build on your own. So we follow the lab life cycles to build the microbiology lab. And so the first steps is to do a community creation of resources. So for that, we developed the Galaxy Codex that collect tools, workflow, and tutors and link them to external repository like bio.tools, Workflow Hub, EDAMS to extract a lot of metadata from these tools and resources. We created the tools, workflow, and trainings available for microbiome analysis, and we improved the annotations of more than hundreds of tool suites on the process in BioTools and EDAMS. So now, within the microbiology labs, we offer more than 880 tools that are grouped into more than 350 tool suites. And they are covering a lot of different, as I say, EDAM operations, and they are available on many different servers. So here are the Use Galaxy Star servers, other public servers where we could get information where are these installed, and we have this information for all the tool suites. We have 150 workflows that are either available on using IWC and Workflow Hub, They are available in workflow, but not from IWC. They are available on public servers, or they come also from the GTM. So that are all the workflow available for the community that the community can really use directly. And then we have 35 tutorials that we developed with the community, covering a lot of different topics and EDAM operation that people could be interested in. Once we have the creations, we can create the lab contents, and build the content is like mostly filling in YAML file and Markdown files. So I will not go too much in details. You can read the publication, the preprint that has been put online last summer about the labs. But yeah, it's mostly using YAML files that give some information and metadata and grouping that together. And so for that, we developed this structure where you have getting started sections, then a focus on microbial isolate with interesting tools, database, workflows, tutorials, and learning pathway that people could be interested in if they are interested in microbial isolate, and we have the same for microbiome. And then we have the sections for community created tools, where we have all the tools available, and that is a semi-automatically filled in. And that covered by grouped by EDA operations, and we have the community workflow with IWC workflows, workflow hub, workflows, GTM tutorials, and other public workflows that are linked directly from the lab there. And this is automatically filled in, or semi-automatically. Sorry, there is still a human creation step at some point. Then all this information is pushed to GitHub, and then they are rendered using the Lab engine, and then we could deploy that on several Galaxy servers. So we deployed it on Galaxy.org, EU, Australia, France, Belgium interesting, Czech Republic interesting, let me know. We can deploy, it's easy to deploy, so we have now the experience with that, so if you want to have it, let me know. We'll be more than happy. And the last steps that for the life cycles is to have a community review and feedbacks. Editing the content, it's something we already have done, so several cycles, and we still do it regularly. So as a summary, so the Microbiology Galaxy Lab is really covering a lot of different types of analysis that are interesting for researchers to do within Galaxy. So that is either interested in ecosystem dynamics or health and disease. We cover antimicrobial resistance. We cover a lot of different types of analysis with a lot of tool suite, workflows, and tutorials. And yeah, on that, I would like to thank a lot the microGalaxy community that is behind this work there. And thank you for your attention. And you can check out preprint, sorry.